Friends, welcome to Biology Made Easy. Hope you are all well. Today in this video, we'll discuss about backcross and test cross, which are also one of two important aspects of Mendel's experiments. So first let us discuss backcross. What is backcross? Usually we know that in a case of monohybrid cross, when a tall plant is crossed with a dwarf plant, so this will be capital T, and this will be small t, and the F1 hybrid will be heterozygous, having one allele capital T, the other allele small t. Now, if this F1 hybrid is crossed with any one of the two parents, that is known as backcross. That means backcross is again of two types. One with the dominant parent and the other is with the recessive parent. So, if it is two types, then number one, that means the F1 hybrid is crossed with the dominant parent. So, what will get? This will give rise to the gametes will be one type, and here this gamete will be two types. That means we will get plants in like this: capital T, capital T, capital T, capital T. Here capital T, small t. Here capital T, small t. That means all plants will be tall. That means you can tell 100% tall. That means when we are making a backcross with the F1 hybrid with the homozygous tall plant, all the plants in the backcross is 100% tall. That's not bad plant. Now let us come to the second one. The second one, the F1 hybrid is crossed with a recessive parent. So in this case, what will happen? The recessive parent will produce gametes of one type, TT, and this will produce gametes of two types. Now, if they will combine randomly, what will happen? Capital T, small t, capital T, small t. This is small t, this is small t, small t. That means here, tall is to dwarf plants. They are 2:2, or we can tell 1:1, or we can tell 50% each. That means the tall plants are 50% and the dwarf plants are 50%, or they are in the ratio of 1:1 when the cross is made with the recessive parent. So this is about backcross. Now the question is, what is the significance of this backcross? Why the backcrosses are made? Now when a cross is made between two plants, what is the intention behind it? That is, two good characters have to be combined together in one parent. So suppose one plant is tall, the other plant is dwarf. When the cross is made, it is tall, but it is heterozygous tall, not homozygous tall. Now, if we want to introduce that tall character in this, so if it will be backcrossed with this tall plant, then it will acquire that character. Suppose one plant has a high protein content, and the size of the fruits or the number of fruits are less. While in another case, the fruits number is more in the plant, but the protein content is less, and we want to combine these characters. So we will make a cross. So the F1 hybrid will be large number of fruits and high protein content. But we want that the protein content should be more. So we have to backcross with that plant having more protein content again and again, so that character will be inserted in that. That is the purpose or significance of backcross. That is one of the reasons of the backcross. The second one, this is heterozygous, and heterozygous plants are usually unstable. If we cross with one of the homozygous plants, then quickly it will become homozygous, so that it will become stable. So these are the two significances. Number one, by backcrossing with, the character can be added more and more. And second one, is it is a quick process of making the plants homozygous. So this is about the back cross. Now let us come to the test cross. There is one difference between the back cross and test cross. In back cross, the F1 hybrid is crossed to any one of the parents, but in case of test cross, it is crossed only with a recessive parent because the name is test cross. So you have to test. So suppose a cross is made between a tall plant with a dwarf plant. So the F1 will be heterozygous tall. And when this plant is self-pollinated, again we will get a tall, and dwarf in the ratio of 3:1. But this plant is homozygous tall. This is homozygous tall, while these two are heterozygous tall. While this is pure, or we can write homozygous dwarf. Homozygous dwarf. Because these two will be capital T, small t, capital T, small t. This will be capital T, this will be small t, small t. So these two are homozygous, this is homozygous tall, this is homozygous dwarf, while these are heterozygous. Now, if you will be given with a plant, suppose you will be given with a tall plant. So, what will be the genotype of that plant? How can you know? That means when a plant with the unknown genotype is given to you, you can know what type of genotype it is by means of test cross. So one of the purpose of test cross is to know the genotype of the unknown plant. That is important. So here the test cross is possible for both monohybrid and dihybrid crosses. So for monohybrid crosses, let us take for monohybrid crosses. For monohybrid cross. So for monohybrid cross, we can cross with this, if we will make a cross with the homozygous, this one. Sorry. If we will make this, suppose one unknown plant is given, unknown tall plant is given, how will you know the genotype? So in that case, you are taking the tall, which is this pure homozygous tall. Now you cross it with the only recessive parent. So what will you get? You will get this side you will get gametes of one type, this side also will get gametes like this. Now this will be capital T small t, capital T small t, capital T small t, capital T small t. That means all plants are tall. That means 100% plants tall. Now this is one. Now come to the second one. Suppose it is given like this, because you do not know whether it is homozygous tall or heterozygous tall. Now you make a cross with this, the recessive one. So what you will get? You will get T, capital T small t. So you say this is capital T small t, capital T small t. Here small t, small t, small t, small t. That means here tall is to dwarf. They are in the ratio of 1:1 or 50% each. So when the cross is made with homozygous, when the test cross is made with homozygous tall, all plants are tall, 100%. But when it is made with the heterozygous tall, the tall and dwarf plants are coming in the ratio of 50% each or 1:1. That is, from this you can know whether the plant that is given to you is homozygous tall or heterozygous tall. Only from the result you can know. Now this is for the monohybrid cross. Suppose we will make a dihybrid cross. So let us make that dihybrid cross on this side. So suppose in one of the dihybrid crosses, suppose it will be dihybrid cross, cross. So suppose in one of the F1 hybrid, suppose capital R, capital R, capital Y crossed with small r, small y. So the F1 will be capital R, capital Y, small y. This is the F1 hybrid. Now when we will back cross it, that means this F1 hybrid, the F1 are capital R, capital Y, small y will be crossed only with a recessive parent, small r, small y, small y. That means now this will give rise to 4 types of gametes: capital R, capital Y, capital R, small y, small r, capital Y, small y. While this will give rise to only 1 type of gametes. So what will be the result you see? Now this will be capital R, capital Y, small y. That means it will be round and yellow. Then this will be capital R, small y, small y. That means this will be round and green. Here you see small r, capital Y, small y. That means this is wrinkled and yellow. And this one is small r, small y, small y. That means this is wrinkled green. That means the 4 types of plants, they are coming in the ratio of 1:1:1:1. This is the dihybrid test cross ratio. So you see, from this, what is the genotype of the plant, easily it can be known. So if we come to the significance of this test cross, number 1 is the genotype of the unknown plant can be easily known. That is number 1. And this test cross was first made by Mendel himself. Until now, it has become a part of genetics. So this is the significance or importance of this test cross. Now let us discuss questions. So one question we can do: which is a test cross? Which is a test cross? A choice. This is one. Then this is one. Then this is one, and the other one is you can write which is a test cross. Now come to the second question. If 2 pea plants with a red, and this red is dominant, with the red colored flowers with the unknown genotype was if 2 pea plants with red colored flowers was crushed with unknown genotype, where in F2 generation 75% red flowers and 25% white flowers were obtained. then the genotype of the parents having colored flowers will be first choice both homozygous, second choice both heterozygous, third choice one homozygous and one heterozygous, and the fourth choice both hemizygous. So you will be able to answer both the questions. Questions are very easy. So thank you very much having an idea about backcross testes and what is significance. Thank you very much. God bless you. Have a good day.